First of all, good morning, everyone, and thank you for joining excuse me, today's webinar presentation. I'd like to first thank Chaeyeon Laboratories, who is our exclusive distribution partner in Korea for MaxCyte instruments and consumables. And I'm very excited to talk with you today about highly efficient engineering of human iPSCs and the derivatives using the MaxCyte Electroporation platform. So previously, I was a postdoctoral fellow in Akitsu Holta's laboratory at the Center for iPSL Research and Application, where we were exploring a lot of different technologies for engineering iPSCs. And at that time, we came across the MaxCyte platform, and I became a customer actually at that time, and I found that it worked actually quite well in iPSC. So we'll be able to share some results with you today, mainly from Akitsu Holta's lab, a lot of the optimization work with DNA plasmid delivery or mRNA, CRISPR, RNP, and SSODNs. And I'll share some other data with you as well, which I'll go over shortly. So this is our disclaimer. And for today's agenda, for those of you who aren't familiar with MaxCyte scalable electroporation technology, I'm just going to give a brief overview of what our platform looks like and what advantages it offers to our customers, and then talk about some iPSL electroporation considerations for your workflow. I know it's not always easy to engineer iPSLs, it can be quite challenging, but hopefully this webinar today gives you some ideas on how you could potentially improve your iPSL engineering workflow. And although we're talking about iPSLs today, this does apply to other cell types as well. So then I'm going to go into some selected data on MaxCyte Electroporation, optimization of mRNA DNA delivery, an example of actually rhesus macaque iPSL electroporation from Shin Kaneko's lab at Saira, and then some examples of how you can utilize SSODN-mediated HDR and iPSCs for making single base pair substitutions. We also have an example from a collaborator delivering DNA transposons into iPSCs for TCR T cells. And then a couple examples of iPSC derivatives, namely cardiomyocytes and neural organoids that some of our customers have electroporated. So with that, MaxCyte was first founded in 1999. We've been doing electroporation for 20 years. We have a scalable electroporation platform, which means we can electroporate from 100,000 cells to 20 billion cells. So we also have an FDA master file, which helps with regulatory filing of your cell therapy product. I will not go into it today, but we have another platform called the VLX, which is even larger scale electroporation, where we can electroporate up to 200 billion cells at 1 liter scale in under 30 minutes. We are listed on the London Stock Exchange, as well as recently on the NASDAQ. And in 2019, we launched our expert series of instruments, which I will go over shortly. So when you think about generating a cell therapy product, it's important to consider one technology from concept to clinic. You need to do a lot of optimization and design of experiments and validation of your transfection at the preclinical stage. What happens is a lot of customers will do this using a platform that isn't scalable or translatable in the clinic, and that leads to a lot of risk and delayed timelines, which overall can cost customers going forward, because it's all about speed. So with the MaxCyte platform, you can go from concept all the way to manufacturing. We offer one platform. We also, I'd like to mention our scientific support for our customers is very much appreciated, in the sense that we work together with you. If conditions aren't working, we'll make suggestions, not just about the electroporation, but before electroporation, after electroporation, and how that can help your results. We do offer scalability and high efficiency. And with efficacy, if you do that reproducibly, you can accelerate your development timelines. So we are the leading choice for advanced cell engineering. We offer a high-performance electroporation platform that accelerates you into clinical translation for next-generation cell therapies. As I mentioned before, we have over 20 years of experience in primary cell transfection. A lot of our customers also do protein production and genome engineering as well. So it's a wide range of applications. And we continue to expand our partnerships, and you can see some of the names below of industry innovators. And there are multiple programs currently in clinical trials. We are also trusted by top academic, translational, and commercial groups, from R&D to process development, all the way to manufacturing. So some of those names you may recognize: genome editing companies such as CRISPR Therapeutics or Editas. Also, with the nonprofit or academic institutes such as NIH, National Cancer Center, Cedars-Sinai, and University of Pennsylvania, for example. We also work with groups, translational academic groups. So this is our series of expert instruments. We have three: the ATX, STX, and GTX. And you'll notice on the left-hand side here, the ATX, it does not have a pump, which is present on the STX and GTX. And basically, that means that this instrument will not do large-scale electroporation, but it does electroporate up to 700 million cells. This is mostly used for basic R&D. The STX is for bioproduction, for customers who are producing proteins, viral vectors such as AEV and lentivirus. And our GTX instrument is a cGMP compliant platform, which you can use for manufacturing your cell therapy product. So instrument features, all four basically have an integrated touchscreen. So the computer and the electroporation unit is integrated in one. It's about the size of a desktop computer tower. We have LED light indicators around the electroporation port, which indicates which stage of the electroporation process you're in. It's very intuitive. All instruments, even if it has the pump for large scale electroporation, can do small scale. So from 15 microliters all the way to 3.5 mLs, that's small scale for us. And the flow electroporation facilitated by this pump can accommodate up to 100 mLs of concentrated cells and loading agent for electroporating 20 billion cells in less than 30 minutes. So the GTX, again, it's 21 CFR Part 11 compliant. It's 21 CFR Part 11 compliant. It has an FDA master file, and it's also cGMP compliant. So in this image, I'd like to just show the range of our, we call them processing assemblies. You might know them better as cuvettes. So going from left to right, you'll see that from the smallest volume, you have, you can go from 15 microliters to 25 microliters with 3 wells to do optimization of your electroporation experiments. And you can go from 75,000 cells all the way to 5 million cells in this volume of electroporation. And if you need to go larger, or if you need more samples, we do have an 8 sample processing assembly for simultaneous electroporation. And what I'd like to point out here is usually customers will optimize at the small scale, let's say 20 microliters. And once you kind of lock in your cell density, which electroporation protocol, and what concentration of like DNA, CRISPR RNP, or mRNA, you take that same concentration when you scale up. So if you go, from 50 microliters to 1 ml, you just keep that same concentration, but increase the volume. So we're able to scale seamlessly from microliters all the way to mLs. And like I mentioned, with the CL2, you can go up to 100 mLs and 20 billion cells. So all of these instruments basically can accommodate the static PAs, processing assemblies, up to the CL 1.1, and only the CL2 can be accommodated by the GTX and STX. So this schematic or illustration, sorry, just basically shows in different colors the different processing assemblies that you can use depending on what your needs are. So with one instrument, you can basically do multiple cell types. Like we have customers using it for HSC, genome engineering by CRISPR, RNP, and also at the same time producing lentivirus, for example, at large scale at over billions of cells. So definitely, I think the flexibility with our processing assembly range gives you a lot of options depending on the cell type and what your application needs are. And our motto is expressing any molecule in any cell. So some of these applications include expressing ion channels or GPCRs, like in HEK cells or CHO cells. More recently, there's a lot of interest in genome engineering applications, especially with CRISPR-Cas9. Perhaps not so much recently with zinc finger nucleases and talons, but definitely with siRNAs, antisense oligos, and more recently with base editors and prime editors. Also, our protein production customers will use CHO cells, for example, to produce antibodies, bispecifics, or fusion proteins. We also have groups using it to express transcription factors or epigenetic regulators, or to establish reporter cell lines. Maybe without stable establishment, you can transiently express the reporter molecule in a large amount and cryopreserve it so that researchers can just take it out when they need it for screening purposes to save time. And lastly, of course, you can use it for viral production, lentivirus or AAV. I have experience using it for virus-like particle production. And so if you have any questions about any of these applications, definitely feel free to contact me or Chayon, and my contact information will be up at the end. This is an important consideration, and I'd really like to highlight. I think a lot of researchers will deliver, in general, plasmid DNA. It's easy to make. Everyone has molecular biology skills, but really, there are certain challenges when delivering plasmid DNA into cells, especially like immune cells, which may detect the DNA as a potential foreign invading pathogen, which induces apoptosis. So it's important to consider what you're trying to deliver into the cell and what kind of effect it can have. It's not just electroporation, but biologically what that molecule is going to do. So if you can deliver, the molecule transiently through mRNA, usually that's gentle on the cell, you can get higher viability and higher transfection efficiency. CRISPR RNPs, for example, as opposed to plasmid DNA, can get into the cell much more easily and with high efficiency. We have customers delivering recombinant proteins into the cell, or even cell lysates. So we do have one customer in Japan who is electroporating tumor cell lysates into dendritic cells. So this slide is just showing different electroporation protocols that we have optimized at MaxCyte. We have over 100 optimized protocols for various immortalized mammalian cells. Commonly, you'll probably know CHO cells or HEK293 or JACAT, for example. We have protocols optimized for mammalian cancer cell lines, A549, K562. We have customers delivering into primary cells, such as keratinocytes, which are quite challenging to electroporate, and we do have an application note for that if anyone's interested, as well as neurons and fibroblasts. And then today's talk is mostly going to go over iPSCs, but we also have a lot of success getting molecules into mesenchymal stem cells, for example. So, I think that is of high interest to researchers in Korea. Of course, with hematopoietic cells, there's a lot of interest in CAR T cells recently, or NK cells. We do have customers working on all of these cell types. And for protein production, we do have customers using these insect cell lines. So if your cell type is not on here, or we haven't worked with it before, we do have 10 general optimization protocols. optimization 1 through 10. So 1 being low energy, 10 being high energy. So if you have an unknown cell type or you're having challenges with your process, you can use these general optimization protocols to kind of get you on track. So going on to today's talk and focusing on induced pluripotent stem cells, otherwise known as iPSCs, they were, of course, discovered by Shinya Yamanaka back in 2007, and initially OCT4, SOX2, KLF4, and MYC were used for the reprogramming using retrovirus vectors. Of course, since then, there have been a lot of developments in the reprogramming fields to reprogram using mRNA or episomal DNA or even Sendai virus, for example. So there are a variety of ways to reprogram your cells. iPSCs have an embryonic-like pluripotent state, which allows them to grow indefinitely. And that's actually very beneficial for regenerative medicine, where you wouldn't have that ability to expand primary cells to a sufficient cell number you need at times. So they can be differentiated into cells, any cell in the body, cells differentiated from the 3 germ layers, and they are very useful for drug screening, disease modeling, and cell therapy. So currently there are over 100 clinical trials being conducted with iPSCs, with 19 being therapeutic, and of course that number is going up. These numbers were taken from a publication last year, so it's likely outdated. So I'd like to go into some considerations for successful iPS cell electroporation. And one important consideration, and I found this from personal experience, is the plate coating being used during culture, and usually that comes with a combination of media being used. Depending on how strongly adherent the cells are to the plate, usually can help predict how robust they are and responsive they are to electroporation energy. So if you're using like a weak plate coating like vitronectin, for example, maybe you'd want to use a lower energy protocol, whereas with something like iMatrix in Japan, laminin 511 fragment, you can probably use stronger energy because they adhere very strongly to the plate. Cell handling before and after electroporation is actually very critical. I know some researchers who scrape the cells to dissociate them before electroporation, but that really sensitizes them to electroporation damage. So you probably want to dissociate them as gently as possible. After electroporation, just from our experience at MaxCyte, we know that recovering the cells for 20 to 30 minutes in a 37-degree incubator before adding media actually really helps with the membrane recovery, which improves the cell viability and the transgene expression. So really, when you're doing electroporation, it's very much a holistic view of, you know, the iPS cells, the quality, how you're handling them before and after electroporation. So the iPS cell clone quality also plays a role in that. You probably want to try to stay under 30 passages, and if the cells are proliferating at a very slow rate, usually they proliferate within 16 hours, then you're probably going to get lower efficiency in electroporation. So there are telltale signs when you're electroporating iPSCs, and you can kind of use some cues when you want to improve your electroporation efficiency. And as I mentioned before, the loading agent type, if you're trying to deliver like a really large DNA plasmid, I'll share some data in a minute, into iPSCs, you can get it in, but probably at lower efficiency than you can get in versus RNA or protein, for example. So a good example of this is just the CRISPR-Cas9 plasmids. They're fairly large, over 10 kb. Usually researchers have to use puromycin to select for cells that have been successfully transfected. It does work, but you can, as most of you may know, plasmid expression of Cas9 can lead to off-target effects in the cell. So you want to minimize that amount of Cas9 being expressed and how long it's being expressed. So really, if you deliver that Cas9 molecule through protein like an RNP, ribonucleoprotein complex, it will get in very efficiently with high cell viability without having to select with like puromycin. So really, I think just starting from the beginning, what are you loading into the cell is an important consideration. And of course, optimized electroporation protocols. We've been working with researchers for a few years now on iPSL electroporation. So we have some good ideas on what works for iPSL engineering, depending on what molecule you're trying to deliver or how they're cultured, and we can definitely give advice on that. So with that, I just like to cover the MaxCyte Electroporation process. It's probably similar to what most of you have done before. Using other electroporation platforms, we would resuspend our cells in MaxCyte Electroporation buffer. I'd like to note that we just have one electroporation buffer stored at room temperature. It's just essentially a salt solution, which maintains good cell viability and electroporation efficiency. So you would resuspend your cells in MaxCyte buffer, add your loading agent, whether that's DNA, RNA, or protein, and just transfer it to the processing assembly, attach it to the instrument, electroporate. Electroporation is pretty fast. And then afterwards, you would carry it for downstream applications. If it's stable cell line establishment, maybe doing antibiotic selection, or we do have customers who, after electroporation, immediately freeze the cells or cryopreserve the cells. So there are a lot of different ways that you can use these cells after electroporation, depending on your application. So now just jumping into the data, this is some data taken from a Kitsuholta's paper in Stem Cell Reports from 2021. This is our general optimization of any cell electroporation. So you'll see energy 3, 6, and 8 here, and 3 is the lower energy, 6 is the medium energy, and 8 is the high energy. And what you'll notice is that the mean fluorescence intensity will increase as you increase the higher energy. So you're getting more RNA into the cell, and that's depicted in this bar graph. So all of the cells are 100% transfected with mRNA for GFP. And I think for you as a researcher, it's up to you to decide, okay, well, 100% expression is good, but how high do I need that expression to be? And if you can sacrifice some cell viability, for example, maybe you, and you only want high expression, maybe you want to go with like energy 8 for the highest expression. But if you need to preserve cell viability and the cell's ability to proliferate and survive, then maybe you want to drop down in the energy to like energy 3. So I wanted to show this just to give you an idea, like when you're optimizing, what kind of things you can look forward to taking into consideration for your workflow. This is an example of plasmid DNA delivery into iPSCs. It's a relatively small plasmid, about roughly 5 kb. So this is showing, again, the 3 different optimization energies from 3, 6, 9. And here you're seeing 2 different concentrations of DNA. So as you go higher in the energy, of course, you're going from like 1% to 34% with the lower DNA. You can get up to 80% with the higher DNA amount using the high electroporation energy. And that's also depicted in this bar graph. So I think, initially, if you try an experiment and things don't work out, there are ways to optimize either by the DNA concentration or by the electroporation energy protocol. Another thing I'd like to call out, and probably a lot of you are familiar with this already, is that the promoter usage in iPS cells or ESCs even is really important. So CMV, here we're looking at CMV promoter versus EF1 alpha promoter. They're both roughly the same size in DNA plasmid size. And this is looking at 200 micrograms per ml, 400 micrograms per ml of DNA. You can see that you can get up to 80% expression with the higher DNA, but the mean fluorescence intensity is pretty low. So CMV is actually quite a weak promoter in iPSCs because it gets silenced easily. If you look at the EF1 alpha promoter, even with half the amount of DNA, you're getting nearly 100% expression. You add more DNA, you're getting even higher mean fluorescence intensity. So again, you can look at the amount of DNA to optimize your expression or the promoter usage in iPSCs. This slide is showing an example of MaxCyte Electroporation with 2 other electroporation platforms. So MaxCyte delivering DNA or mRNA for GFP, you can see that there's a nice balance of both viability and efficiency. You get pretty good expression, whereas Platform A gives you the viability, but variable expression. And you can kind of see that in the flow cytometry plots on the right here, where it's very heterogeneous. Platform B gives actually quite good expression, but nearly all of the cells die. So, and you can see that here in this bar graph, where the cell viability drops 24 hours after electroporation. So with that said, usually we see either or with other platforms. Maybe you get the viability but no efficiency, or you get the efficiency but no viability. With MaxCyte, you can get the best of both worlds. So definitely, I mean, consider not just the efficiency, but the viability and what you need to do with those cells after electroporation. Earlier, the examples I showed were with about a 5 kb plasmid. Here, we're looking at a large DNA plasmid expressing GFP. This is about 10.3 kbs. And we were trying to optimize electroporation protocols for larger DNA. So the protocol that was used for the smaller DNA, that's number 1 here, gives like 64% expression. But we really had to kind of revamp parameters, and we found with newer protocols that we can get up to 96% expression of even large DNA. So we have tweaked protocols to efficiently deliver larger DNA as needed. And I think this slide's really interesting. This slide was reproduced with the permission of Stephen Sloan from Emory University, and also Raphael Kroll's group at the CSIRO Foundation. But what you can see is up to 10 kb, at least, we're getting pretty good expression of the plasmid with a reporter gene, either GFP or mCherry. And as you go larger in the plasmid size, you're going to see a considerable drop, excuse me, in the electroporation efficiency. So up to 16 kb, and I know a lot of researchers will go up to 16 kb because they're trying to put just a lot of components into this DNA vector. You're dropping down to 20%. And if you're really trying to use that 16 kb vector as a DNA donor template for CRISPR knock-in, you're going to expect even lower knock-in efficiency. So just wanted to point out that, you know, the size of your DNA vector when you're delivering it into cells does matter. So if you can potentially reduce that size, you can increase the amount that's getting loaded into the cell. The next slide here, it's an interesting slide where we're looking at the co-delivery of either large plasmids of 16 or 12 kb with about a 9 kb plasmid. And basically, you can see from the 10.3 kb plasmid, that's kind of MaxCyte's control, you can get like 90% expression. This is customer plasmid that was about 9 kb. We're getting like over 90% expression. If you co-express the or co-transfect, sorry, the 9 kb with the 16 kb plasmid, we don't see any color from this plasmid B because it only expresses if you do something to it later. So basically the expression of the smaller plasmid does drop a little. But I'd just like to point out with in this situation here where you're kind of seeing a 9 kb with a 16 kb plasmid, you can co-deliver them roughly at 10%. In the case where the plasmid is a little bit smaller, like 12 kb, you can get over 50% co-expression of mCherry and GFP. So just wanted to point out here, you're probably limited by the larger plasmid delivery into the cell. So if the larger plasmid gets in, most likely the smaller plasmid will get into. But I know there are a lot of researchers who are trying to co-deliver larger DNA plasmids. So I just wanted to demonstrate that it can be done, but it is a little bit more challenging as the plasmid gets larger. For customers who are looking for scalability, I just like to kind of call out here that we electroporated 5 million cells in our OC100 processing assembly. So in about 50 microliters, and we could get about 78% of GFP expression 2 days later. If you take, like I said before, if you take those same conditions of the cell density and the DNA concentration, we could scale up to 175 million cells. And this is our CL 1.1 processing assembly that can go up to about 3.5 mL. So you're going from 50 microliters to 1.75 mLs, and you're seeing very similar GFP expression. So really depending on the need for how many cells you need to electroporate, I just wanted to show this, that even with iPS cells, you can get really good scalability from low microliter range up to over an ml of electroporation volume. The next example here is taken from Shin Kaneko's laboratory at Saira. So this was published in a paper in molecular therapy methods in 2021. But in this paper, they were taking rhesus macaque iPSCs, and they were struggling a lot. I think they struggled for over a year with other platforms and trying to get not only efficient transfection efficiency, but importantly, knockout efficiency. So just some optimization experiments initially. They didn't really optimize. They used the same electroporation protocol used for human iPSCs for these rhesus macaque iPSCs with MaxCyte, and were able to get 93% expression of GFP mRNA versus 64% with another platform. Also, the viability was better. But the genome editing was the important parameter. They couldn't get any homozygous knockout clones, and they're trying to knock out TRIM5 alpha here because I think they were looking at HIV susceptibility studies. But here, in the first try, they were able to get homozygous clones that they could take forward for their experiments where they were stuck for a year. So I think that efficiency, of course, is higher now. They've been able to optimize a little bit better, but I just wanted to point out it's not limited to just human iPSCs, but also non-human primate iPSCs. Next, I'd like to just kind of go into monogenetic diseases caused by single point mutations. The majority of monogenic diseases are caused by these single point mutations. So researchers doing disease modeling, for example, will either take a healthy patient iPS cell line and genome edit it to mimic the disease model, or if you have access to disease patient iPS cells, you can correct the mutation to generate an isogenic clone for drug screening purposes, for example, or just disease modeling. So with this in mind, SSODN delivery is actually a very efficient way to moderate or mediate the HDR base pair substitution in iPSCs. So here you're looking at an example, and this is also taken from a Kitsu-Holta's paper of the DMD gene. So they're in this example using a SSODN template that's about 100 base pairs in combination with CRISPR-Cas9 protein and guide RNA, basically making 2 base pair substitutions from A to C and A to T, which introduces an AG1 restriction site. So the purpose for this was to induce exon skipping to restore the dystrophin gene expression once iPS cells are differentiated into skeletal muscle cells. But before doing that, we did optimize the knock-in efficiency of this SSODN. So you can see that with varying concentrations of the SSODN, you can get over 60% knock-in efficiency. And I think this is pretty important because in this case, no small molecule HDR enhancers were used, no temperature shifting. It was just electroporation alone. So basically, this data demonstrates that out of 10 clones, you should have at least 6 with the desired HDR knock-in of your SSODN template. And this slide is just showing a comparison with MaxCyte and the 4D nucleofector. This is also published in Akitsu's paper, where you can get a certain extent of knock-in efficiency with the other platform, but the variability was much higher. Whereas with MaxCyte, it was fairly reproducible and with high efficiency over 60% consistently. So we also wanted to point out that, comparing MaxCyte with the 40 nucleofector, this is just looking at the relative cell number. I believe in this case, it's 48 hours after electroporation. But just looking at electroporation alone, for example, you're seeing like a dramatic decrease in the cell number with another platform. With MaxCyte, there's a slight decrease with the cell number. With DNA, especially like SSODN, you're seeing a really reduced amount of cells, much lower viability. You still kind of see that with MaxCyte, but to a certain extent, the cells are surviving better. So because of these results, we kind of speculated that the higher cell viability might be leading to higher HDR frequencies or gene editing efficiencies. And so I'll show in this next example where Miyoshi myopathy patient iPSCs were engineered. So again, iPSCs are an essential tool for disease modeling and drug screening. And in these cells, there's a single nucleotide polymorphism which introduces a premature stop codon. So to generate an isogenic clone for basic research, the goal here was to electroporate CRISPR-RNP with a 100 nucleotide DNA donor template, and basically to convert the stop codon to arginine. And so again, this is just converting the stop codon to arginine. What you can see here on the right figure is that with MaxCyte Electroporation, with two different concentrations of SSODN, you're actually seeing an increase in knock-in efficiency over 70%. Whereas with other platforms, and we've seen this with other platforms too, where the higher you go with the amount of DNA, you start to see a reduction in the knock-in efficiency just because the cell viability is much lower the more DNA you put in. So here, just highlighting that you can get high knock-in efficiency, and you can titrate your DNA donor template to achieve your goals. So just wanted to kind of focus in on this data set here. We did take about 43 clones from these bulk edited cells and performed genotyping on them, and found that 73% of these clones were homozygously corrected. So which is pretty impressive, again, considering that no enhancers were used or other ways to boost the HDR editing. The next example I'd like to show is how you can use SSODNs to generate heterozygous HLA-C iPSCs by electroporation. So as I mentioned earlier, iPSCs have a lot of potential for regenerative medicine, but if you have a homozygous donor iPS cell line, it is still susceptible to NK cell cytotoxicity. So actually a PhD student at the time in Akitsu's lab, Huaiging Zhou, came up with a strategy to generate HLA heterozygous iPSCs. And the way to do this was to deliver multiple SSODNs into the cell at the same time. So this is kind of what's going to happen if you use a homozygous donor iPSC, which mismatches with one of the KIR receptors. You'll have cytotoxicity. The goal here was to modify one of the HLAs to HLA-C type 2 and basically promote cell survival. So in this example, in allele 1, you're still keeping the C1 type, but you're introducing a silent mutation so it doesn't get cut again by CRISPR-Cas9. And then you're mutating these two amino acids into asparagine and lysine for C2-like KIR. Or HLA-C, sorry. So this is basically showing the results where we're getting the expected silent mutation introduced, as well as the two other mutations to induce the HLA-C conversion. So in this case, it was about 1 out of 29 clones for simultaneous knock-in of both SSODNs, but I think this data highlights that you can perform complex engineering of iPS cells for the goal of allogeneic transplantation. This schematic just shows shortened timelines using MaxCyte Electroporation. At least this was what we did in our laboratory, where we electroporated cells with RNP and SSODN. You would check the knock-in efficiency at day 3, but at the same time, you're kind of expanding the cells until day 10, where we would single sort them into 24-well plates, and then expand them. And then by day 30, we would be able to freeze and check the knock-in efficiency by Sanger sequencing. So within a month, you can get your desired clone of iPSCs with the correct base pair substitution by SSODN and CRISPR-RNP electroporation. And I know there are longer approaches which involve, incorporating an inducible Cas9 into the genome by piggyback and then excising it later. So that can take more than 3 months and it's extremely laborsome. So this is a pretty straightforward approach, which can be incorporated into your current lab's workflow. Just quickly, this is a poster that we generated in collaboration with Dias. Dias is a startup company based in Kyoto and is a spin out of Shin Kaneko's technology for iPS cell derived T cells. So in this poster, basically the researchers were using piggyback DNA transposon to deliver into iPSCs to stably express a TCR molecule. They would differentiate into T cells using embryoid bodies and then look at both in vitro and in vivo cytotoxicity models. So early on the optimization, they found that if you express GFP DNA plasmid, you can get like 90% expression. After 14 days, this is not integrated, it's only transiently expressed. So the expression drops to 0%. And then with the DNA transposon, they were getting 82% at day 2. And by day 14, they were getting stable integration of about 47%. So using these cells, they found that the ability to differentiate into hematopoietic progenitor cells, and this is what different TCRs transduced into the iPS cells, was not affected. So electroporation did not affect their ability to be differentiated into hematopoietic progenitor cells and subsequently T cells. And they continue to use these TCR expressing T cells in an in vivo model, which were intraperitoneally injected with SK-HEP1 liver cancer cells expressing luciferase. So you can see with the control, the luciferase expression increases. There's nothing suppressing the proliferation of these cancer cells with 4 different types of TCRs. And you can see it by the measurement of chemoluminescence. You're getting suppression of the luminescence signal. So indicating that in vivo, these T cells are functional in expressing TCR to suppress the cancer cells. And next, just going into the iPS cell derived or derivatives, sorry, these are iPS cell derived cardiomyocytes, and this data was generated by in collaboration with ACE-RNA, and actually the researcher was a good friend of mine, Shinil Kim, who is now the CSO at Therabest. So he generated this data where he was looking at just GFP mRNA transfection into iPS cell derived cardiomyocytes. And basically what he saw was using 3 different energy protocols, kind of similarly to what I showed you before with different energies, you can get 100% expression of the mRNA. Viability was also very good. It's just a matter of the MFI that you need for your application. But this clearly demonstrated that MaxCyte Electroporation was feasible for iPS cell-derived cardiomyocytes. And the last piece of data I'm going to show is with neural organoids. This was done in collaboration with a laboratory at UCSF. So for any organoid groups out there, using neuronal organoids, we electroporated them with membrane-bound GFP-expressing plasmid. The organoids were between 0.5 to 1.2 millimeters in diameter, and with a couple of different optimization protocols on our instrument. So we looked at GFP expression 1 day after electroporation, and then also stable GFP expression 3 to 5 weeks post-electroporation, looking at the penetration of the plasmid DNA into the deepest layers of the organoids without causing dissociation. So here what you're seeing is day 1, day 4 expression of GFP in these neural organoids, and this is using 2 different electroporation energies, 5 and 7. And basically you can see long-term expression of GFP 3 weeks and 5 weeks after electroporation by spinning this confocal microscopy. So again, this is with permission from David Rayleigh at UCSF. So with that, just going into the summary, we can help with highly efficient transfection of difficult to engineer cells such as iPSCs, a lot of flexibility with the type of cargo you'd like to deliver into the cell. We have high cell viability post-electroporation, and for genome editing in HDR frequencies, that usually tends to result in better editing efficiencies. The higher editing efficiencies will hopefully lead to lower cost, less labor, and shortened timelines for your lab. And it's always a rush to publish, of course, so faster timelines are better. We have a one-step biallelic engineering process using SSODNs, and also just wanted to point out that we can efficiently electroporate iPSC derivatives, including neurons, cardiomyocytes, and neuronal organoids. So with that, I hope you got an idea of different ways you could potentially use MaxCyte Electroporation for your experiments, or maybe different ideas on how to improve your current engineering workflows. So thank you for your attention. I'm happy to take any comments or questions. You can contact me directly, or you can contact our distribution partner, Chayan Laboratories. Thank you.